Hello everyone, and welcome to our lecture on inheritance and development. So when we talk about genetics, we talk about all of the genes that make all of our proteins that do all of the work in our body, right? And all of the genes that we get come from our parents. So inheritance is the way in which we acquire the genes from our parents, and genetics is the systematic investigation of the study of inheritance. We as humans have 23 pairs of homologous chromosomes. It means we have one pair of chromosome 1, one pair of chromosome 2, one pair of chromosome 3, etc., all the way up to one pair of chromosome 22. However, chromosomes set 23 are going to be sex chromosomes. So ladies, you have two X's. Gentlemen, you will have one X and one Y. And each of these chromosomes are going to come as a single chromosome from your father and an equal and opposite chromosome from your mother. These are called maternal and paternal chromosomes. And genes that are going to be for the same trait don't have to necessarily be the same identical gene. So if we have variations within that, those are called alleles. And alleles generally arise from mutations that have come up previously. So a mutation is defined as a permanent and heritable change in an allele. And that means that it's going to get passed down from parent to offspring. Okay, so an example of a genetic disorder is something called PKU, or phenylketonuria. And if you have this disease, PKU, you're unable to make something called phenylalanine hydroxylase. And phenylalanine is an amino acid, and you need to break down phenylalanine, and if you're unable to do so, you can get extremely sick. And one way that we look at the way that we acquire genes from our parents is called a Punnett square. So we use the Punnett square in order to look at the possible outcomes of the way genes are going to be inherited from two parents. So let's talk about a little bit more nomenclature for a second. So we have two major ways that we can look at inheritance patterns. So we can look at genotypes and we can look at phenotypes. So genotypes are when we're actually looking at the genetic makeup. So we're looking at two alleles that come together in the genotype. And those alleles can be either recessive or dominant. So a dominant allele is going to be the one that is going to mask the presence of the alleles around it. And recessive alleles are only ever going to be seen if there's two of them together. So there's not a dominant allele present to mask the recessive allele. And so a phenotype is what happens when we put those two alleles together. So that's the actual physical expression or outward characteristics of the individual that has whatever the genotype of interest is. And this here is a Punnett square. You've probably seen this in high school biology. So basically what happens if we look on the upper left here, here's the father's options for the chromosomes that he can pass on to the sperm. You can see here that he's depicted as being heterozygous. So he has one dominant allele and one recessive allele, and in just genetic nomenclature in general, the dominant allele is going to be depicted in uppercase and the recessive allele in lowercase. So this father is able to make two different sets of sperm. Remember, in a gamete or a sperm or an egg cell, you're only going to be passing on one copy of each of your chromosomes, even though the individual has two. So that means that they're going from what's called a diploid state, having two copies, into a haploid state, right, just having one copy. And that means that this sperm can either be carrying the dominant allele or the recessive allele. Now, if we look at the mother is also heterozygous in this instance, so again, she can create two different sets of ova. Half of those ova are going to be carrying the dominant allele, the other half are going to be carrying the recessive allele. And mathematically, either of these sperm could interact with either of these eggs. So we have to prepare for all four of those possibilities. So that's why we're going to go ahead and make this Punnett square here. So here on the left, we can see what happens with the male sperm. So again, the male is able to either donate the dominant allele, or in this case here, the recessive allele. And then the mother, the same thing. She can donate either the dominant or the recessive allele. And then when you bring those together, so for example, if this egg and this sperm meets, we're going to have two dominant alleles coming together. So we're going to get this genotype here, dominant, dominant. If, however, this egg and this sperm meet, we're going to have two recessive, so coming from both parents. Here we have the option of having a heterozygote if we end up having the dominant ova meeting up with the recessive sperm, or vice versa. So we end up getting a ratio here if we're looking at the genotypes of 1 to 2 to 1, right? So basically going to be 25% dominant, dominant, 50% heterozygote, and 25% recessive, recessive. But that's going to be what happens if we're looking at the genotype. Now, if we're looking at the phenotype, however, all three of these individuals which have that dominant allele are going to have the dominant phenotype. So while we have a genotype of 1 to 2 to 1, we're going to have a phenotype of 3 to 1. And in this case, this unfortunate individual here, which has that 2 sets of the recessive phenotype for PKU, are going to end up affected by this disorder. This is just a list of different traits that humans have and the way that they are acquired in a hereditary fashion. So things that are dominant and recessive, and this is only for very, very basic things like whether or not you're able to taste something called PTC, which is going to have a bitter taste on your tongue, and whether or not you have the correct amount of digits, etc. But there's going to be a lot of alternate ways in which things can be inherited, so that's kind of oversimplifying things if you're talking only about the dominant recessive inheritance patterns. We also have incomplete dominance, so basically what that means is that the alleles, instead of one being completely dominant over the other, they're going to kind of push back, so you're going to get an intermediate phenotype for those heterozygotes. And a good example of that is something called sickle cell anemia. So if we end up with the heterozygote of sickle cell anemia, actually confers a little bit of resistance to malaria. Okay, so this is another Punnett square. So again, you've seen these before. This is the father's options, and then after meiosis we're going to be creating sperm, and then the mother again creating these eggs. So these are the possibilities of two individuals that are heterozygote for sickle cell anemia, and that means that they're able to contribute either the normal allele or the sickle cell allele to their sperm or to their ova. And when those guys meet up here, we are going to end up with that same thing that we saw previously, where we have 25% are going to be homozygous for the dominant version, which is going to be the normal version, and then here we have 25% that are going to be homozygous for the sickle cell version. And unfortunately, these individuals here are going to suffer from sickle cell disease, and without regular transfusions, they're going to die. So in a first world country, they will have the options to survive, but generally in a third world nation, this individual would not live to lead a happy, normal, healthy life. But what's interesting here is that these individuals in an area that is prevalent to malaria, they could end up having a high percentage chance of succumbing to that disease state. Whereas this heterozygote individual here and here, so 50% of the offspring, are going to end up with a little bit of a conference of a resistance to malaria. All right, so another way that we can have an inheritance happen with other inheritance patterns is something called multi-allelic inheritance, where multiple alleles are involved. And sometimes these alleles can have a difference of the way that they interact with one another. So, for example, I know we talked extensively about blood typing previously, but in the example of blood typing, we have two different sets of the way these alleles interact with each other. So A and B are going to be normal codominant, so they're going to be expressed equally, whereas A and B are both going to be dominant over O. So an individual that has an AO genotype is going to have the phenotype of an A individual. So either AO or AA will give you the same outward expression or phenotype. So this is kind of an overview. It is an overview of what can happen if you have parents that, in this case, both parents are blood type A. And if you are blood type A, that means that you can have a genotype of either A and A or A and O. So in this case, that means that the offspring that can arise from these individuals, it can either be A or O. If, for example, both of these guys are heterozygote AO, they could potentially both pass on the O allele, and the offspring could be an O offspring. If, however, the individuals are A and B, again, they could still have that hidden O allele in there. So this individual could be A, or she could be A, O. This individual could be B, O, or B, B. So in this way, we could inherit multiple different combinations. So the individual could end up being A, B, or O, depending on what they inherited from each of the parents. Okay, so I won't walk you through all of these options, but basically what happens is the way that the genes interact with each other is going to be different whether or not you're talking about how A interacts with B, or whether you're talking about how A or B interact with O. Another inheritance pattern is polygenetic inheritance, and that's when we have multiple different genes that are controlling the effects. And we can see Additionally, on top of that, the environment can also play a role. So we can have the combined effect of multiple different genes and environmental factors. So when I originally showed you that dominant recessive options, it's a very oversimplified view of what's actually going on. Most of the traits that we actually see in the individuals are very complex traits. That's going to include things like skin color, hair color, eye color, height, intelligence, body build, metabolic rate, etc. So most of the things that we characterize ourselves by are actually a complex combination of all of the different genes that contribute to that trait. And this is an example of an inheritance pattern where many genes are going to be responsible for the contribution. So in this way, we actually have depicted 6 different genes. And they can all be either homozygous dominant, heterozygous, so one big A and one little a, or in this case over here, it can be homozygous recessive. And we can do that for either A, B, or C. So in that way, when we have that next offspring, they're all going to be intermediates. They're all going to be heterozygote for A, B, and C. And when individuals that then end up with heterozygosity breed, then we can end up with a whole combination of ova and sperm that can vary from all of the lightly colored genes to all of the darkly colored genes, depending on which combination they get. And obviously, I'm not going to walk you through all of these combinations here, but this is just an example of a more complex inheritance patterns. Okay, in humans, we have 23 pairs of matched chromosomes. There's 46 of them total. So in a diploid individual, which is what all of your somatic cells, so every cell that is not a sperm or an egg, you're going to have 46 chromosomes. However, in that specialized set of cells, the sperm and the egg, there's 23 individual chromosomes. And we're able to identify them by multiple different ways, by their size, their shape, their staining patterns, etc. And we can look at them in what's called a karyotype. So a karyotype is when we take all of those chromosomes and we lay them all out, and we do this according to size and where the centromere is. And that centromere, are we going to show us one? They are. So the centromere is going to be right here in the center. So depending on where the location of the centromere is, and you'll see some of them are going to be evenly matched approximately, and some of them are going to have much smaller and much longer arms. These are called P and Q regions of these chromosomes. And what they've done here is they've laid it out by size. So chromosome 1 is the largest, down to chromosomes 22, which are the smallest. What's interesting here is this is the X and the Y, and the X is much, much larger than the Y. In fact, the Y really only carries genes that are involved in male differentiation and development. If you don't get the induction of the male differentiation cascade, you become a female, but that X has a lot of very important things on it, and this is why some things are going to be inherited in a pattern of male-female, because, for example, gentlemen, in your sons, you are only ever going to be able to pass on that Y. So that X never gets passed on from a father to a son. So certain things that end up being inherited, that the mother is a carrier, but the sons end up affected, might include things like color blindness, for example, male pattern baldness, etc. So there's a lot of very important things that are actually carried on that X chromosome. And ladies, that's why one of them is actually shut down in every one of your cells in your body. So preferentially, one X is going to be shut down in every cell of your body, because we don't want to end up with double the expression of the genes that are on the X than the men do. Alright, so again, males, you have one X and one Y. Females, you have two X's. One of them is going to be automatically turned off. That's called X chromosome inactivation. And once it's turned off, it's going to become something called a bar body, which is visible in that karyotype. Now, the gender of the offspring really is decided by which sperm fertilizes the egg. So if the sperm is a Y-bearing sperm, that egg is going to end up becoming a male offspring. If that sperm is an X-bearing sperm, it's going to yield a female offspring. And that's the real way that gender is determined. So because an egg is only ever going to have an X chromosome in it, because it was derived from a meiotic event from a cell that had two X chromosomes. So there was never a Y in the picture. So this is what that looks like during the meiotic event. So instead of having what we previously saw as depicted as dominant or recessive, we're still just looking at alleles here, or entire chromosomes here, I apologize. So this father, who's an XY, he's able to either create an X-bearing sperm or a Y-bearing sperm. And this mother, she's only going to be able to create an X-bearing ova. So when we put that together, as you see here, the eggs that are fertilized by the X-bearing sperm are going to become females, and the eggs that are fertilized by the Y-bearing sperm are going to become males. And that's how we end up with approximately 50-50 percentage gender of male and females in the offspring. Now, some things are inherited on that X chromosome, as I mentioned previously, and anything that's inherited by way of the X chromosome is called sex-linked traits, and this is why more men in the room are going to be red-green colorblind than women in the room, and that's because, ladies, if you have one bad copy of an X that doesn't allow you to see color, that's the one that's preferentially going to get shut off. Men, you guys don't have that backup, so you are going to be what's called hemizygous for this trait. That means that you're carrying whatever you're carrying on the X, and then you're carrying the Y. So you only end up with one option, which means that if you're carrying that trait that doesn't allow you to see color, you've got that mutated allele, unfortunately, you're not going to have anything as a backup to compensate. And that's what this looks like. So if here you're looking at C would be the abnormal color, this is color vision. So if we have the capital C, that's going to be normal color vision. The lowercase c is going to be abnormal color vision or color blindness. And a female who is going to have one copy of the normal color vision and one copy of the abnormal ability to see color vision, again, color blindness, she would actually be able to see color vision just fine because this normal copy would compensate and this one would be preferentially shut off in all of the cells in her body, or at least in the cells that were needed for processing color vision. However, males, over here, if you were to receive one bad copy of the non-ability to comprehend color, you would end up colorblind. So let's look at what that looks like if we're looking at a Punnett square. So this is that mother who is a carrier. She has one bad copy of the color vision gene. That means that she can make two different subsets of eggs, one that carries the X for normal color vision and one that carries the X that will lead to color blindness. Right? If an X-bearing sperm from the normal male that is able to see normal vision is going to come in, then we have two different sets of daughter offspring that can be created, right? This daughter offspring will have both normal vision alleles and will have normal color vision. This one will replicate what is the situation of the mother, where having one bad copy here and one good copy means that she is a carrier because she will be able to see color vision, but her offspring are at risk, particularly her male offspring, and this is why. So when the Y-bearing sperm comes in to fertilize, now this male, well, both of these males are what's called hemizygous. They have one copy of this allele and they get what they get. So in this case, this X is going to have the normal color vision. So this male offspring is going to have normal color vision. And this one is going to have the abnormal color vision option and therefore is going to have color blindness because there's no backup mechanism to compensate for that. All right, so let's shift for a second to talk about development. So we have a couple definitions that we're going to talk about here. First, we are going to talk about the fertilization event. The fertilization event is what happens when the sperm and the egg are going to actually meet, and then the cell's going to divide. The embryonic period is going to extend from that point, so from the fertilization event forward, through to about the 8th week of development. After this, it's called a fetus. So the way that the fertilization event occurs is that sperm are going to swim. They actually have flagella, so a little tail that's going to take them from the site of deposition in the vagina through the cervical mucus, through the uterus. They're going to go all the way up to the uterine tubes, which is going to be the site of fertilization. Okay? In order for the sperm to be able to get to the point where they fertilize the egg, they have several barriers. Again, we talked about the cervical mucus. They have to swim through the uterus, and those of us who were here for my reproductive lecture, I went into that in great detail. But one of the things that they have to do is get through the granulosal cells, which are called the corona radiata. And that's going to be surrounding something called the zona pellucida, and the zona pellucida is basically a tight layer of glycoproteins that's directly outside of the oocyte's plasma membrane. Okay, so what does that look like? And you may remember that this is going to be the cumulative effort of many different types of sperm, because these sperm have a little region on top of their head here called the acrosome, and that acrosome contains specific enzymes. Those enzymes are going to be secreted to help penetrate through this, what's called the cumulus mass, to get through the corona radiata to get to the zona pellucida. Okay, once one sperm gets through the zona pellucida, the whole thing becomes an ironclad lockdown so that no more sperm can penetrate. Some other things of interest here is going to be the first polar body. This is going to be the other results of the meiotic event that produced this oocyte here, and then plenty of cytoplasm in here. And you'll notice that the sperm has almost no cytoplasm at all. All of the cytoplasm that's going to contribute to the developing embryo from this point forward is going to come from the maternal lineage. Okay, so as I mentioned, there's an acrosome that's found on top of the sperm's head. It has a bunch of enzymes inside of it. Those enzymes are going to be released that are going to help the sperm get through that cumulus mass that's called the corona radiata, and also get to the zona pellucida, where they're going to bind with a specialized protein that's called zona pellucida binding protein 3, and that's a receptor whereby the very first sperm is going to bind, and then all of the rest of the proteins around the zona pellucida are going to enter into lockdown. This is to prevent a secondary sperm from fertilizing the egg. So basically what happens is we have the sperm that are releasing these enzymes from the acrosomes, and then they are going to digest through this mass of cumulus cells, getting to the zona pellucida to bind to ZP3 to be able to be the first one to penetrate through to get to the one that fertilizes the egg. So this is the head of a sperm cell here. This is the tail. This is the oocyte. So this is what it's going to actually be. This is the one that's making contact. Here's the zona pellucida. So the head of the sperm has a nucleus inside of it. Remember, it's haploid. So unlike the rest of the cells in your body, your sperm and your egg only have one copy of each of the chromosomal normal pairs. So normally we have 46. Again, the sperm and the egg are going to have 23. That makes them haploid. And the haploid nucleus of the sperm and the egg are going to come together. So the male and the female pronucleus are going to come together. That's called syngamy. And that's the actual process of fertilization. That's when the diploid zygote is formed. So we have restored our genetic complement by merging together the genetics from the maternal lineage and the paternal lineage to create a genetically unique individual who is genetically unique both from all of its siblings and also from both of its parents. All right, so these are the male and female pronuclei. They are meeting inside the egg. So the sperm has actually come through, it started to decondense, and now we are going to put these guys together. This is the actual act of fertilization, you guys. So for about 24 hours after fertilization, we're going to undergo the first mitotic division, and that's called a cleavage event. It takes about 6 hours. The ones that occur from this point out are going to take less time. So this is the 2-cell zygote. It's at the approximately 1-day stage. In fact, it's still surrounded by that zona pellucida, right? That zygote is going to continue to divide until here's a 4-cell stage, and we're going to then continue dividing into 8, 16, etc. At the end of the 3rd day, we have 16 cells. And what's interesting here is that this size is actually remaining approximately the same. So as you know, we have 2 cells. Now we have 4 cells that are a little bit smaller. Now we have 16 cells that are much smaller. And so the moriula is going to maintain its size until a certain point. It's approximately the size of a mulberry. Again, the zona pellucida is still surrounding it, and it is still the same size as the original zygote. So we haven't gotten into a serious growth event yet. At this point, you have to remember the fertilization event occurred up in the uterine tubes or in the fallopian tubes, and it's been rolling down, this moriula has been rolling down into the uterine cavity until it can find a happy place to implant itself. As it does this, it's going to be nourished by the uterine fluid or uterine milk, which are going to be secreted out from these endometrial glands, as well as feeding off the nutrients that it stored previously in its own cytoplasm. Okay, once we get to the 32-cell stage, it's going to start shifting around. So no longer are all of the cells going to be everywhere. They're starting to orient themselves. And we're starting to get a section that's going to be fluid filled. That's called the blastoquele. That's going to have a blastocyst cavity. And the mass itself, the mass of cells itself is called the blastocyst. And again, it's still approximately the same size as that original 2-celled zygote. Now from the outside, the blastocyst is just going to look like a large mass of cells. But inside, we have 2 separate sets of cell populations. We have the inner cell mass and the outer cell mass. And the inner cell mass is called the embryoblast, and it's a precursor to the embryo. The outer cell mass is called the trophoblast, and it's eventually going to contribute to the accessory structures, including the chorionic sac that surrounds the fetus, and the fetal portion of the placenta, which is called the chorion. All right, so if we were to take a section of the blastocyst and take a look through it, the internal view would show us that we have an internal cell mass that's oriented itself in one direction, and the blastocyst cavity inside. Again, this whole outer section is the trophoblast. So the blastocyst is still going to be rolling around the uterine cavity for the first 2 days or so, and eventually will find a nice place to implant itself by attaching itself into the endometrium. This happens approximately 6 days after the fertilization event. Again, these are the endometrial glands that are going to be secreting that uterine milk that are going to help sustain the blastocyst, and is also, again, still surviving on its own nutrients as well. So then implantation has to occur, and that generally is going to occur in the posterior portion of the fundus, or sometimes inside the body of the uterus itself. There are specialized regions that are going to be best sites for implantation. So a high-risk pregnancy might be a pregnancy that is implanted in a region that is not as suitable for the best fetal development and growth. And that inner cell mass is going to start orienting itself towards the endometrium. It's going to be burying inside the endometrium shortly. So again, here's our endometrial glands. These are going to be secreting that uterine milk. Here's the maternal blood supply and blood vessels, etc. This is the embryoblast. That's that inner cell mass. Surrounding that is the trophoblast. Remember, this is what's going to become the embryo, and then this is going to become some of the supporting structures. And inside here, we have this whole cavity region that's called the blastocyst cavity. And we're about to be embedding ourselves into the endometrium. So after the implantation period, the endometrium is now, instead of being called the endometrium, it's now called the decidua. So basically, an endometrium that is supporting a pregnancy is changed to become a decidua. And that's actually going to be coming out with the fetus, or immediately after the fetus is delivered. It's going to separate off from what would normally be considered the endometrium. And the decidua has multiple different regions, and it's all named based on how they are relatively oriented to the implanted blastocyst here. So here we have the implanted embryo. It's now gotten underneath that layer. Here's the decidua basalis that's going to surround that implanted embryo. And then the region on top is called the decidua capsularis, I apologize. And then the region that surrounds that is called the decidua parietal. All right, as I mentioned, the fertilization event actually is happening inside the uterine tube. So let's recap. The sperm have to make it all the way through a lot of different barriers to fertilization. First, they have to make it into the uterus. Then they have to make it all the way up into the uterine tubes, and then they have to get through that cumulus mass, bind to the zona pellucida, and fight their way through. Once the fertilization event occurs, then that zygote is going to be tumbling. And it's going to be undergoing cleavage events as it goes through. Its first 30 hours or so are going to be spent in transit through those uterine tubes. Now it's going to be called that morula. Remember, that's about 3 to 4 days after the fertilization event. And eventually it's going to find itself in the uterine cavity as a blastocyst. This happens approximately 5 days after fertilization. It still hasn't implanted. It has to be able to find a happy place to make its home and then bury its way into the endometrium. And that's going to happen approximately 6 days after the fertilization event. About 8 days after the implantation event, that trophoblast that we talked about is going to develop into 2 different regions. It's now going to become the syncytiotrophoblast and the cytotrophoblast. And that embryoblast is also going to split into 2 separate layers. These 2 separate layers are going to be the endoderm and the ectoderm, and later on they're going to end up adding a mesoderm. Currently, though, we only have 2 layers, and again, the embryoblast splits into the hypoblast, which becomes the endoderm, and the epiblast, which is going to become the ectoderm. And these are going to make a flat disc. It's bilaminar. Again, we have the endo and the ectoderm, so we have a bilaminar embryonic disc. It's going to become trilaminar again in just a moment, well, in a few days when we're going to add in the mesoderm. And the amniotic cavity is going to form from the region that was the epiblast region. Okay, so now we're at 8 days after fertilization. Again, endometrial gland. This is the endometrium itself. We are working our way in currently into the endometrium. Here's the trophoblast. The trophoblast is split up now into 2 separate regions. The cytotrophoblast, which is this region here, and the syncytiotrophoblast. Also, points of interest here, this is the epiblast, which is going to line that amniotic cavity. This here is the amniotic cavity, and then this section is actually the amnion. We are also going to be making something called the exocolemic membrane, and that's forming directly here, coming from this hypoblast region. This section right here is that bilaminar embryonic disc I was talking about, and again it's split up into the hypoblast, which is shown here in yellow, and the epiblast depicted here in blue. In the center here we have the blastocyst cavity, and the whole thing again is going to be implanting into the endometrium, so the side is the uterine cavity and the side is the endometrium itself. So then amnion is going to actually be forming on the roof of the amniotic cavity, and eventually that's going to be filled with that amniotic fluid that we're very familiar with that's surrounding the entire embryo during development. Approximately on the 8th day, that region that I just talked about, the exocolemic membrane, is going to form entirely, and that's going to form what's called the yolk sac, and that forms from the hypoblast region and the exocolemic membrane. And that yolk sac is going to be very important. It's going to be helping to both feed the developing embryo and also going to become a very important part of the developing embryo, and we'll talk about that when we get to the invagination steps. Approximately day 9, we have something called lacunae that are going to be forming. Lacunae is just a word that means small spaces, and eventually those are going to be forming together to form what's called a lacunar network, approximately on day 12. Alright, so again, reorienting ourselves. This is the endometrium of the uterus. We have now completely successfully embedded ourselves into the uterine lining. This here is the amniotic cavity here. And again, we still have that bilaminar embryonic disc, which is separated into the epiblast and the hypoblast. And then around that, here's the exocolemic membrane right there, which is going to directly connect with the hypoblast and form what's called the yolk sac. So this region here is going to be the yolk sac. And then what else do we have here? So then the outer region here is what was the trophoblast layer, which is now split up into 2 sections called the cytotrophoblast layer. That's depicted here in pink. And then outside of that is the syncytiotrophoblast section, which is going to actually be directly interacting with the endometrium of the uterus. So we just went back. We're at 9 days. Now we're at 12 days after fertilization. We are entirely embedded in the endometrium. These are the lacunae that I was talking about earlier. They're going to form the lacunar network when they are all going to end up binding to one another and forming these spaces, and these basically sinusoids are going to connect with them. So it's going to allow an interaction between the maternal fluids and the developing blastocyst. Again, in yellow, here's our yolk sac. That yolk sac is going to line right up on the opposite side with the hypoblast region of the bilaminar embryonic disc. Inside, shown in blue, is the epiblast, again the amniotic cavity and the amnion itself. And now we've added something new here. This here is the extra embryonic mesoderm. So as I mentioned previously, this is a bilaminar disc for now, and it will very soon become a trilaminar disc with the mesoderm coming in to form the 3rd layer. Okay. Okay, so about 12 days after fertilization, we're going to make that mesoderm that I just talked about. That's the extra embryonic mesoderm. And we're also going to have a region that connects around the amnion and the yolk sac, and then making those extra large cavities that are going to form. Those are called the extra embryonic column. So this mesoderm is going to be very important. It works in concert with the trophoblast to form something called the chorion. And the chorion is going to surround the embryo and then later on the fetus, right? Because as soon as we end up losing the yolk sac, it becomes a fetus instead of an embryo. The chorion serves multiple different purposes. The first thing that it does that's very important is it makes human chorionic gonadotrophin. And we're going to talk a lot about hormonal regulation in a minute, but the HCG is basically what you test for in your pharmacy pregnancy tests because it is what helps sustain the pregnancy. We also are going to need the chorion to help reduce the antibody production by the mother, because the mother is now in a situation where it has foreign matter in her body. It's non-self, but it's a non-self that we don't want to attack the way we would normally want to attack something that would be non-self in our immune system. And along with that, we want to suppress our immune system, and we do that by promoting the production of T lymphocytes, specifically to suppress the immune system in the uterus, but it does also have a global effect on the maternal body. Approximately 3 weeks into development, we have a very large event called gastrulation. And gastrulation is very important because we are now taking that mesoderm and putting it right in the center of what was a bilaminar disc and making it a trilaminar disc. So now we have the ectoderm, mesoderm, and the endoderm. And this is going to happen quite quickly, and it's going to happen with a mass rearrangement of different cells. So they're going to migrate from different regions of the developing embryo. And the first step of this is the formation of what's called the primitive streak. So this is when we first start making orientation, right? So we're starting to orient the head region and the tail region. This here, just to kind of orient us, is the dorsal surface of the bilaminar embryonic disc, which again in a moment is going to be a trilaminar disc. The bottom section is going to be that yolk sac. So basically we have the yolk sac and the amnion separated by this bilaminar embryonic disc that is comprised of the epiblast on the amnion region and the hypoblast on the yolk sac region. And what we've done now is we've established orientation. So we've established the head and the tail sections of the developing embryo. And now we're going to undergo what's called invagination, whereby we're going to be moving cells inward underneath that primitive streak to pull in that mesoderm. So we're going to create 3 germ layers. And that looks kind of like this. So that mesoderm is going to just be forcing its way in between the ectoderm and the endoderm. All right, so we've changed our terminology. So let me go back just a second, right? A second ago we called it an epiblast and a hypoblast, right? Now we are calling it an ectoderm and an endoderm, all right? So these are still the same cell layers. It's just that now we're orienting things a little bit more in a little bit more of a complex fashion. We also, again, we have this primitive streak here, and that's going to be oriented against the oropharyngeal membrane, so that's going to be the head side, and then the part behind us is going to be the tail side. I am not going to go through all of the different structures produced by all of the 3 germ layers, but I do kind of want to point out some overviews. So basically the endoderm is going to be creating the inside. It's going to create the inside of our GI tract, the inside of our bladder, the inside of our lungs, and so the epithelial lining of almost everything that's going to be internal is going to come from the endoderm. The mesoderm, again the middle, is going to make cartilage, bone, connective tissue, the lower layers of the skin, the kidneys, the internal organs, the gonads, right? So all of the stuff that's in kind of the middle of the body. And the ectoderm is going to be the external stuff, including our nervous tissue, the epidermis of the skin, a lot of things that have to do with our eyes and our ears and our sense organs, and then pigment cells and a lot of the skeletal and connective tissue. So again, outside. So endoderm, inside, mesoderm, middle, ectoderm, external or outside stuff. Around 16 days, we're going to start making something called the notochordal process, which is eventually going to become something called the notochord, which is going to be where we are eventually going to be making our spinal information, right? So notochord is going to be very important for making tissue that's going to respond to all of the different structures around it. And it's going to induce the development of things called vertebral bodies, which are going to be prevertebrae, basically, and the nucleolus pulpus of the vertebral discs. So the notochord is eventually going to become the spinal column. All right. So here we have a dorsal and a sagittal section of our trilaminar embryonic disc. So basically what's happened now is we have forced our way in here with our mesoderm. So now our yolk sac is actually going to be separated from this section here, the amnion, by what's now the mesoderm. We've also oriented ourselves such that we have the head region, the oropharyngeal membrane is going to be the head, and the tail, which is also known as the cloaca. So this is the cloacal membrane. So it's going to be the opposite side. Cloacal will be associated with the tail, oropharyngeal associated with the head. Now underneath this ectoderm, it's outlined by this little depiction here of the dotted lines. That's going to be the notochordal process. And this here, this little peanut-shaped thing, is going to be the neural plate. This region here, primitive node and primitive streak, and again, all of this is going to be part of the invagination process, which is going to happen very shortly. All right, so some other structures are going to be created here. Again, that oropharyngeal membrane. the cloacal membrane and the alointus is also going to be created. And that alointus is going to be a connecting stalk from the yolk sac, or connected to the connecting stalk that comes off of the yolk sac. All right, so the development of the neural plate is going to be stimulated from the nodal cord region. Here we have this notochord section right there. This is the neural plate, which is about to undergo a process of invagination. Again, the neural plate is going to be lining the amniotic cavity when we have here the layer of the ectoderm, and then on the inside middle layer, the mesoderm, and then the endoderm lining the yolk sac. Now, as I mentioned, we're going to start the neural fold, which is eventually going to become a neural an entire invagination. So here are these cells that were a second ago flat, and now we have a neural groove and a neural fold on either side of that. We also are going to have these guys here are going to have a little bit of invagination as well, and again, this is the notochord. So this little invagination that I just talked about 1 second ago here, these little red invaginations, those are eventually going to become, I apologize, the somites. And somites are going to be developing alongside this neural plate. And this is here is going to be the neural groove, and eventually this section there is going to be on top of that as the neural crest. All right, so as the neural folds are going to fold into one another, they make what's called the neural tube. The neural tube is going to run along the entire embryo, and that process of invagination and formation of these structures is something called neurulation. I also want to draw your attention here to these somites. We now have more somites that are occurring. The somites are actually going to be a way by which you can age, or stage, I guess, the developing embryo, and they are eventually going to be part of the vertebral column. Let's see, so what are we showing here? Let's talk about the somites for a second. Here, these are somites. These somites are created of 3 subsections, the derma, myo, and sclerotomes. Derma, myo, and sclerotome region. And this is also part of that intermediate mesoderm. So this is going to be coming out of that middle section, the mesoderm. So that neural tube is going to invaginate, and then on top of that, what was previously ectodermal cells are going to migrate to form a layer of cells, or a couple layers of cells, called the neural crest. So that looks kind of like this. So this region here is the neural plate region, which is eventually going to become the neural tube, right? We're going to have neural folding that occurs, and this section here and this section there are actually going to come in contact with one another to become the neural crest. So we are going to have an invagination event occurring that brings this section to that section across the top there to become the neural crest. And then inside there, we're going to have, so this section here is the neural crest, and then underneath that we're going to have the neural tube. Again, the notochord, and this is the endoderm. So let's talk about the brain for a second. The head region is going to start developing the brain around 4 weeks of fertilization. We're going to get these specialized structures that are going to be called primary brain vesicles. And there's 3 major ones that are each split up into their own sections. This here is the forebrain. It's the prosencephalon. The mesencephalon is the midbrain, and the rhombocephalon is the hindbrain. So the rhombocephalon is where we're going to have the ear developing, and it's going to lead down into the spinal cord and spinal column. And this region here is going to get split up. Remember, just a second ago we talked about that being the prosencephalon. It's going to get split up into the diencephalon and the telencephalon. And the midbrain is just going to be that mesencephalon. Here in the rhombencephalon, here in the hindbrain, we have the metencephalon and the myencephalon. And again, that leads down into the spinal cord. And don't worry, guys, I'm not going to quiz you too heavily on all of the different regions of the developing embryo. We're just kind of walking through the process here. So as I mentioned, we have those somites. Somites are paired structures. They are on either side of the spinal column, basically, and we end up with approximately 42 to 44 pairs of them by week 5. So you can determine the age of an early embryo based on the presence and location of the somites. Now, the somites are going to differentiate into 3 different regions: the myotome, the dermatome, and the sclerotome. And those are going to go directly around, here's the neural tube, around the developing nervous system. Again, just to orient you, here's the notochord right here in the center. And we're starting to end up with the imagination of the large blood vessels. This region here is going to become the cardiac region, so the future aorta, so the top of the heart. All right, so speaking of cardiac, let's start talking about blood vessels. So the creation of blood vessels is called angiogenesis. And the way that these are made is that we start with something called blood islands. These guys here are individual blood islands. And they have precursor cells here called angioblasts. And angioblasts are going to lead to the creation of blood cells. So we're starting to get orientation whereby we have this region here of the endoderm, we have the mesoderm, and eventually we're going to end up with lumen, or little space that's going to surround this angioblast that are then going to create blood cells. So what we've done here, if you can consider that this is a hose that's opening up, this is becoming the lumen of the blood vessel. So we've actually done a kind of a cross section through it, and inside here we now have blood cells. So the blood cells are developing at approximately the same time that the blood vessels are developing. So when circulation kicks in, it's all ready to go. So the heart's going to start to develop about day 18 and 19. And it develops in a region called the cardiogenic region. These are made of mesodermal cells. And the first thing that happens is we have cardiac tubes. There's two of them, so it's a pair of these endocardial tubes. And they fuse together to create a primitive heart tube. And we talked about this a little bit when we talked about the cardiovascular lecture. But basically what happens is in this head region, we have the cardiogenic area. And that's going to end up creating these endocardial tubes, which are then going to fuse in 4 different regions to eventually become these bulbs. And then we're going to be able to start having blood flow. But we eventually are going to have to fold over upon ourselves a couple of more times to get our finalized cardiac positioning. And here we can see this is our normal adult setup here with the aorta and the pulmonary trunk and all of the different ventricles. There are a lot of things that are going to be different, of course, for the embryonic and developing fetal heart. We will talk about that once we talk about post-birth and what happens. But there's going to be a little hole in the center here that allows blood flow back and forth between the two atria, such that such high pressure that may end up happening is not going to tear the developing tissue, and that eventually closes once the heart becomes strong enough to be able to sustain that pressure. Okay, so the embryonic tissue is going to invade into that uterine wall, right? And eventually it's going to be getting rid of the uterine blood vessels, and then we're going to end up with blood filling into those lacuna spaces. So if it wears down the uterine blood vessels are going to start leaking and filling into these lacuna spaces. At the same time, we have chorionic villi that are developing, that are going to be on the fetal side, that are going to be helping with the maternal-fetal exchange of. So the blood itself does not exchange, but matter is exchanging between the fetal and maternal blood supplies, or will be eventually when we end up making the placenta. Okay. So this here is the maternal sinusoid. Again, a sinusoid is just kind of like a hole that's going to allow fluids to leak through, right? So fluids are going to be leaking into this intervillous space. This intervillous space is going to line right up with that trophoblast region, which has now become the syncytiotrophoblast, here depicted by this like yellow-brown area. And the cytotrophoblast is depicted here by pink. So this area is the intervillous space whereby fluid can leak from the maternal sinusoids out into this region and then go across into the region that surrounds the blood capillaries and then can get picked up for the embryonic circulation. So this is a way that we can have maternal exchange of things that are not going to be blood cells themselves, but are going to be things that are going to be nutrients going in and waste going out. Eventually, this is all going to connect to become the umbilical cord. So the vessels are going to connect by way of umbilical arteries and an umbilical vein through a body stalk, which eventually connects to become the umbilical cord. Okay, so currently, the fetus is still, or the embryo, I apologize, the embryo is still feeding off of this yolk sac, but eventually we're going to be creating something called the placenta, and then the yolk sac will not be necessary anymore, and that will be the way in which we are going to have maternal and fetal exchange of materials. Now, once that yolk sac is going to dissipate out, and we'll talk about what happens to it in just a minute, and we have a replacement of the placental, then we are going from an embryo to a fetus. So that is when we are going to change our terminology. And speaking of the placenta, the process of creating the placenta is called placentation. And this is also going to be a region where we're going to be exchanging materials, things like nutrients and waste between the mother and the fetus. It's also going to create hormones, so it has a series of hormones that it's going to be secreting for the rest of the pregnancy. And it's actually considered an organ. It's a temporary organ that's created only in certain times of necessity, like pregnancy, obviously, but it is considered to be a organ. So the placenta has two different regions: the fetal portion, fetal region, and the maternal portion. And the fetal portion is going to be what I just showed you with the chorionic villi that extend out and are going to be able to pick up things that are, or exchange wastes. And the maternal portion is going to be from the decidua basalis of the endometrium. And we talked about that when we talked about those sinusoids that were being created and how they were going to be leaky. So this is kind of what that looks like. This here's the yolk sac. The yolk sac is still feeding this developing embryo, and it's going to be doing that by way of this alointus here, right? So that's going to be connected here at the yellow. But we also have in the formation of the umbilical cord, which is going to be connected eventually to the placenta. On the outside here, these are the chorionic villi. This is where we're going to actually be having exchange of materials. This is going to be fetal associated tissue. It's actually fetal tissue. It's going to be fetal and maternal associated tissue. And this is what that looks like. So this is the maternal portion of the placenta on the left. This is the fetal portion of the placenta here on the right. Again, the maternal portion is in the decidua basalis of the uterus. The fetal portion is going to be in the chorionic villi and the chorion region. Again, that looks here. So we've basically taken this little box here and pulled it down here and looked up close. So on the maternal side, we have venules, right? And we have arterioles that are feeding into these regions that are just giant open intravillous space where the blood is going to pool in and then pool out. So it's going to basically leak in and leak out. And then across this barrier here, which is the fetal portion of the placenta, here we have these villi that are going to have blood vessels inside of them. Right, it's called a chorionic villi, and things are going to be able to leak across that membrane and also be secreted out into that maternal blood, where it will then be, so the waste of the developing fetus will then go out through that endometrial venule. So once we finally have a fully developed placenta, it's going to be quite large and pancake shaped. And it also has an additional role in protection of from microorganisms, because the fetus is going to be susceptible to anything that may end up in that amniotic fluid. So the very first line of defense is going to be having to get through the maternal blood supply to be able to get into the fetal blood supply. And so it's going to be basically the first line of defense for the fetus. So between the 4th and the 8th week, we have a very large push of organogenesis, or the creation of all of our major organs. And approximately week 4, we have that embryonic folding event that I've spoken about a little bit. We'll talk about it a little bit more. And basically what happens is we take what was previously a flat embryo and we fold it up, or kind of roll it up into a 3-dimensional cylinder. So you can picture taking a tortilla and making it into a burrito, except obviously a lot more complicated. The first thing that has to happen is we have to have invagination, little folds that develop at the head region and the tail region. Okay, so this is going to be the head end and the tail end, and we're going to have a little bit of fusion that's going to form here at the oropharyngeal membrane. We're going to have the same thing happening here at the cloacal membrane. Again, that's going to be oriented towards the tail of the developing organism. This region here in the center here, that's the notochord. On top, we have the amniotic cavity, and the bottom, the yolk sac. All of this should be very familiar to you at this point. Now, this here, again, that's going to be the notochord. Above that, the neural tube. And then above that, this region is the amniotic cavity. This section here is going to be the coelom, the intraembryonic coelom. And then these guys here are going to turn into somites, etc. So first we have lateral folds that are going to form, and once these lateral folds form, then they're going to move inward towards the midline, and they're going to incorporate that yolk sac as the primitive gut. So I told you that yolk sac wasn't entirely going to disappear, right? It's just going to kind of get folded into the developing embryo and become the route by which food enters the body and waste exits the body. So it's going to become the entire digestive tract. There's a section that's outside the embryo that is the little cavity, and that's called the proctoduum. That's here. And this is basically going to be the area where the tail fold is going to meet up here with the hindgut. So this is going to become the midgut, and then this is the head fold, just to kind of orient you. This section here is the intraembryonic coelom that we spoke of previously. Here's the invaginated yolk sac, and again with the neural tube, and the section there being the amnion and neural crest. So what separates the cloaca from the proctoduodenum is that cloacal membrane, and that's right here. So here's the proctoduodenum. Again, it's what's going to be connecting this region here of the developing yolk sac, and that's the cloaca, which is again towards the tail, and that cloacal membrane connects those two together. Here we have the same thing occurring on this side. This is the oropharyngeal membrane, which is going to be connecting this region here called the stomotoduum, which is opposite of the proctoduum, and that's going to be the head section connecting again the oropharyngeal membrane. We have regions called the pharyngeal arches, or also known as the bronchial arches, and they're going to develop on each side of the head and the neck during that 4th week when we have a huge push for organogenesis. And each arch is separated by a pharyngeal cleft. So these are the pharyngeal arches. They're separated out by the pharyngeal cleft, and they are eventually going to become the throat. So the pharyngeal pouches are going to extend out here and eventually meet those pharyngeal clefts and the pharyngeal folds. It's going to become the throat, and then underneath that the esophagus. You can see this here is the developing lung bud, so that's going to become part of the respiratory tract. And then this section here is going to be the neural tract, so the brain, etc. So if we're doing a transverse section here through the pharynx, you can see that each one of these little pouches has everything you need to be able to create everything you need, right? Cartilage, nerves, muscles, and so each of these arches are going to eventually become the pharynx itself. Also around that 4th week, a lot of things are happening. We're starting to develop the limb buds. We're getting the upper and the lower limb buds and something called the heart prominence, which is going to be where the heart is going to be located. It's going to be a little region of the area pouch where it's going to be basically making room for the heart to grow. Also at the end of that 4th week, we're going to have a tail. We're going to sustain that tail for a good bit of the pregnancy. Eventually it's going to undergo apoptosis, and we are not going to carry it on into the full-term pregnancy. Around week 5, the brain and the head are going to continue to rapidly develop. The limbs are going to further develop. Remember, we previously just had limb buds. Now we have hand plates and foot plates. Again, these are the pharyngeal arches that are eventually going to become the throat. We have a second prominence here, this liver prominence, which is going to pouch off. It's going to be the region where obviously the liver is going to develop into. Around week 7, we end up with fingers and toes. So we end up getting distinct limbs and distinct digits. However, we're still going to have webbed fingers, so we will not actually have specific fingers until the end of the 8th week. So around the 8th week, we end up with eyelids that are going to come together. We're losing that tail, and we're going to start the differentiation of the external genitalia. So if we are a male, we're going to be able to start possibly being able to tell the sex of the individual around week 8. Our digits are going to be distinct at this point. We have lost all of the webbing in between our fingers and our toes. We're also going to start seeing, instead of just seeing like arm buds, we're actually going to start seeing distinct regions of the arm and the development of early bones, so early ossification is going to be occurring as well. But mainly what's going to be here is all going to be cartilage at this stage. From this point on, we're basically going to be undergoing a lot of growth and differentiation. So we're not going to be making a lot of new structures until much later. Basically what we're going to be making, and the next structures that we're going to be making are going to be things like fingernails and eyelashes, etc. So very few new structures are going to appear up until much later on. During this time, we're basically just undergoing a lot of growth and differentiation from 9 week on. So this is showing you the 20-day embryonic stage to the 24-day to the 32-day, etc. So as we're working our way forward. So here we are just getting to the point where we have the orientation. We have the neural plate, the neural groove. We still have the yolk sac feeding, so we have not even created the placenta. Eventually at the 24-day stage, we're going to start getting major prominences like the heart prominence coming out, the somites, etc. By 32 days, we're going to get what looks to be an actual embryo with distinct regions like the limb buds. Again, we have the tail. Here's the lower and the upper limb buds. We're going to have those pharyngeal arches, which are going to be the pretty classic early embryonic stages. Area where the eye is going to be coming in, called the lens placoid. The otic placoid will be coming in a little bit later. Here's the 44-day embryo with the otic placoid. This is where the ear is going to be coming in. And we're getting very distinct upper and lower limbs at this point, and also very shortly going to start seeing things like fingers, right, actual digits. So around the 10-week fetus, you're going to start seeing things like the actual fingers appearing. Around 13 weeks, you're going to see all of the digits and things like the eyes, the ears, the nose, and the mouth. Much more detailed structures are going to be forming. And again, from this point forward, we're just going to have a lot of growth and differentiation and development. So we're not going to see anything new appear for quite a while. So this table here is going to have a summary of all of the things that we've basically told you. So the embryonic period lasting from 1 to 4 weeks is when we're going to have the notochord developing and neurulation occurring. We're going to be creating the early structures like the primary brain vesicles, the somites. We're going to start creating the blood vessels, and also going to start forming the heart and get that beating heart. And placental formation. Embryo is going to fold into itself. It's going to create the pharyngeal arches, the gut and the limb buds, etc. So everything's going to basically be setting the stage. From the 5 to 8 week stage, we're just going to basically be getting more specialized. The limbs are going to become very distinct. We're going to see the fingers, 4-chambered heart now, as opposed to previously we just had those heart folds, and also going to start having the eyelids fusing together, the nose are going to be developing. We're going to start having identifiable human-like characteristics, and bones are going to start forming. Mainly we're going to have cartilage forming, but we're going to be laying down the framework for where the bones are going to be in place. Right? Then from this point forward, from the 9th week forward, we're going to enter into the fetal period, and this is basically what happens when the placenta takes over. And each of them are going to be marked by several major events in major organ systems. I'm not going to go through all of them, but basically each of the organ systems is going to go through developmental stages that occur at each of these major time points. Oh, another thing that's important to note here, right here is when right around here is when we're going to start having fetuses that are able to survive if born prematurely. From the 24-week mark on, now obviously early fetuses can have serious complications, including having issues with vision and hearing and immunocompromisation. Obviously they're going to have to stay in the hospital for quite a while, but from this point forward, they're able to survive without the assistance of the mothers in terms of getting their own nutrients, etc. Previous to this, we're going to be talking about a loss event if the mother ends up in something fatal like a car accident. So this is what the overview of the size of an individual as we head over. So we're going to get larger and larger and larger, and also get more and more distinct features, including the digits, eyelids, and eventually getting things like hair and eyelashes. So if a compound, generally a chemical compound, is going to cause issues with the development of an embryo or a fetus, it's labeled a teratogen. And these are things that pregnant women are encouraged to avoid at all costs. Now, teratogens can be chemicals that we use routinely, but are fine for normal adult individuals. So there's a ton of things that are normal chemicals and or drugs that are fine for adults, something like alcohol, for example, but can interfere with the process of development of major organ systems in an embryo or a fetus. Some other things, they don't have to be chemical. They can include things like viruses, hormones, antibiotics, other drugs like cocaine, LSD, etc., and many, many others. There are a lot of different sets of fetal birth defects that are kind of classically associated with certain problems or certain things that the mother has done during pregnancy, including cigarette smoking or fetal alcohol syndrome. So cigarette smoking during pregnancy has a whole list of defects, of presentations, including low infant birth weight, sometimes can have some cardiac abnormalities. Anencephaly is issues with the brain. Anytime you see cephaly, it's going to be brain issues. So hydrocephaly would be water on the brain. Anencephaly would be a lack of a brain, or microcephaly, small brain, etc. Also, high infant and fetal mortality rates because of just the inability of the small developing liver to be able to handle these toxins that are being put through it. Another teratogen is ionizing radiation. So this is why women during pregnancy have to be very careful about how often they go in for things like x-rays or exposure to radioactive isotopes during certain medical procedures. That might be fine for a normal female at her age, but is not okay for the developing fetus. So oftentimes one of the first questions they'll ask you when you enter into a hospital, at least as a woman of reproductive age, is, are you pregnant? And oftentimes they're going to go ahead and run that pregnancy test, even if you say you're not pregnant, just to be sure, because they want to make sure that they're not in any way causing any harm to developing embryos. So a lot of issues can arise if you subject yourself to a teratogen while you are a pregnant female. So we have ways of testing for multiple sets of fetal abnormalities, as I just demonstrated, and we really just kind of glossed over how difficult the processes of all of these major steps are. But anything can go wrong in the early stages, and we use modern medicine to detect abnormalities and genetic disorders, and also the overall well-being of the fetus throughout the early pregnancy and development. In fact, some things that are fetal abnormalities or genetic disorders that might be a problem if they were left untreated are things that we can now treat. So sometimes it's really just knowing that there is a problem and being able to avoid it. So oftentimes these are going to be things that are going to be able to be easily dealt with. So we use a lot of tools. One of the tools that we use is a sonogram, right? Fetal ultrasonography, and that's going to be oftentimes used to determine accurate fetal age as well. Sometimes women don't know exactly when they had conceived, and oftentimes also they'll just go by the date of the last menstrual cycle, which gives you a little bit of leeway in terms of when you could have gotten pregnant within that window. We also use ultrasound to confirm pregnancy, to demonstrate that you really are pregnant and you don't just have some other underlying problem that causes pregnancy symptoms. And also determines the fetal position and identifies multiple pregnancies, and can also be used to identify other issues that might be a problem, like uterine fibroids, for example. So we use it for a lot. We also use something called amniocentesis, and amniocentesis involves using the amniotic fluid. So they isolate the amniotic fluid that surrounds that developing fetus, and they analyze it. And they use the fetal cells to check for genetic abnormalities. Usually they do this between 14 to 18 weeks. It's a little more dangerous as you get further along in a pregnancy, because you have to use a very long needle, and although they do guide it by ultrasound to make sure that you're avoiding either the umbilical cord or the fetus itself, the larger the embryo, or at this point the fetus, the larger the fetus, the less room there is for error. So this is what that looks like. This needle is going to go through the abdomen, through the uterus itself, and collect fluid from the amnion, or from the amniotic fluid, and then use that to spin it down. They'll isolate it for fetal cells, and then use that to test to see, well, several different things. But generally they're going to use it, particularly in the advanced maternal age, to eliminate the risk of something like Down syndrome, which would be trisomy 21, which is fairly easy to detect if you were able to actually get fetal cells, which they get from the amniotic fluid. We can also take cells from the chorionic villus sampling, and they go directly basically into those chorionic villi and pull samples out. They can do that as early as 8 weeks of gestation, and again they're going to be using an ultrasound to guide that catheter in there. They take a catheter and insert it in through the vagina, and then through the cervix, and then up into the chorionic villi to collect a tissue sample. And basically it's the same goal as the other test. They want to check and make sure that we don't have specific genetic defects, and it can also be done through the abdominal wall if they don't want to, for some reason or another, go through the vagina itself. And so this is basically just showing you how we would do it. We would use an ultrasound to determine where everything is orientation-wise. They use a long catheter to go through the vagina and then head up into the uterine cavity and collect some of the chorionic villi. And again, they could also do that by going directly through the mother's abdomen with a needle, but that involves piercing through the uterus, which is just an additional risk that most women don't take if they don't need to take. We have a ton of other prenatal tests that we use that are considered to be non-invasive. However, we are not able to get as much information out of them, so they're a little bit less informative as something like amniocentesis and chorionic villus sampling. But of course, all of this is optional, and mothers are given the information that they need to make decisions on whether or not they want to take the very small risk to themselves and to the fetus in order to have the information in their hands to be able to make decisions that they may need to make. Another way that we're able to check on the status of the pregnancy is by checking on AFP. And maternal AFP levels are going to be something that would be indicative of a neural tube defect if they are too high. So this would be something that might indicate that you have an issue with neurulation or with spinal column formation. And an incomplete spinal column formation can be known as spina bifida, which is something that can happen if a woman doesn't get enough folic acid during her pregnancy. So anyway, if a test comes back that we have inappropriate levels of AFP, this might be a difficult pregnancy, and that female might be headed into some very difficult decisions. So this might be something where the attending physician would want to monitor very closely. All right, so if you guys have been paying attention to my reproductive lecture, then you would know that the corpus luteum is the region of the follicle that is left after it bursts open and releases the ova. And it's left in the ovary, and generally it takes about a week or so, 2 weeks to degrade. Meanwhile, it is secreting progesterone and estrogens in low levels. However, in the case of pregnancy, there is a signal that's given to that corpus luteum to continue to secrete the progesterone and estrogens, and it will do that for about 3 to 4 months until the placenta takes over. So the corpus luteum is actually going to be kind of the surrogate for the placenta hormonally for the first 3 months of pregnancy. Now, the chorion is going to secrete human chorionic gonadotropin. And you may have heard of this because this is what's in all of our regular over-the-counter pharmacy-style pregnancy tests. And basically what happens is the chorion from the embryo is going to secrete HCG, and HCG is going to be the trigger that stimulates the corpus luteum to continue to produce these estrogens and progesterone far longer than they normally would, thus inhibiting menstruation, and it will be able to do this again far longer, normally only about a week or two, and here it's able to do it 3 to 4 months because of the HCG that's secreted by the chorion. So what does that kind of look like? Okay, so HCG, again, this is what's found in all of your ELISAs of your pregnancy tests. This is going to be a hormone that is going to affect the corpus luteum in the ovary and prevent it from degrading very quickly. So normally it would degrade in a matter of weeks. Here it's going to remain for 3 to 4 months. And it's going to be secreting progesterone and estrogens during this time frame. Ladies, those of you that are on the birth control pill in the room, this is generally what the birth control pill mimics, are higher levels of progesterone and estrogen, which is going to then delay the body's reaction, which would normally be menstruation, and then from ovulation for the next month. And so basically it tricks the body into thinking that you are already pregnant, and so therefore you're not going to release another egg, and so you're not going to get pregnant from that point forward because the body thinks it already is. Anyway, so usually if you are checking your birth control pill, it's going to have levels of progesterone and estrogen that are going to help maintain the endometrium of the uterus, mimicking pregnancy. Additionally, it's going to be responsible for the creation of, or the preparation of the mammary glands, which are going to create milk during a process called lactation that comes from a hormone called prolactin, and is also going to help change the body of the mother in ways that are going to help her prepare for delivery of the baby. Additionally, the corpus luteum secretes something called relaxin. Okay, relaxin is also secreted by the placenta, so the placenta secretes HCG, which then affects the corpus luteum secretes progesterone, and also relaxin. The placenta also secretes relaxin. Relaxin is involved in making your joints very flexible, and this is going to be important because we're going to be making our pubic symphysis actually distend such that we're able to push a baby through something that is normally much, much smaller. And so this is going to help us with the dilation of the uterine cervix and also with the stretching of the pubic symphysis. However, some of the side effects of this can include things like hyperflexible ankles and elbows. So people may end up, pregnant ladies later on may end up having to wear knee braces because they might have a trick joint that pops out of socket or something like that because they have a high level of relaxin in their body. Another hormone that is secreted by the placenta is called HCS, or human chorionic somatomammatrophin, and that's going to help prepare the mammary glands for lactation. So it's all in preparation for giving milk to the new baby. It's also going to enhance the growth by increasing the amount of proteins that are synthesized, and also going to increase fatty acid use for ATP production and decrease the glucose use by the body. So all of this is going to be helping get the body prepared for delivery. Also in terms of the infant, enhancing the growth of the infant itself. It also secretes corticotrophin releasing hormone. And CRH is going to help dramatically with establishing birth timing. So it's going to be very important in labor and delivery, and it's also something that sometimes you can have added if you are having a birth that is not progressing properly. It also is going to increase the secretion of a cortisol, which is going to help with stress levels, etc. So it's a very stressful time. You kind of want to stay a little bit relaxed throughout it. This is an overview of different hormones, not all of the ones that we talked about, but some of the important ones, including HCG, estrogen, and progesterone, and when they are typically secreted in the human body during the embryonic development. So HCG levels can be detected as early as 2 weeks. This is why people really love that over-the-counter pregnancy test. It can be detected before you're even supposed to be menstruating. And in the early stages, they ask that you wait to collect your morning urine because you can concentrate the amount of it in your urine. And that's because in those early stages, we have such a small amount in it that it might not necessarily be detectable yet. So you might have a negative test and then next week have a positive test because next week your levels spike to detectable levels. And then for the next, up until week 8 or so, week 9, the HCG levels are going to be maintained at high levels. Again, that's coming from the chorion. That's why it's called the chorionic gonadotropin. It's secreted by the chorion, and that's going to tell the corpus luteum to continue to secrete estrogens, right? So we also are going to be secreting estrogens from the chorion approximately 3 to 4 weeks in, and then progesterone is going to come in approximately the 6th week. From that point forward, progesterone and estrogens are going to be the ones that are going to help with the maintenance of pregnancy up until birth itself. So HCG is actually going to drop off in those moderate, about the 2nd trimester or so. Relaxin is going to be secreted both by the corpus luteum and also by the placenta, later on by the placenta, early on by the corpus luteum. Remember, the corpus luteum is only going to have a major role for the first 3 to 4 months, and then once the placenta takes over, the corpus luteum is just going to continue to degrade. But relaxin is important in creating flexible joints, including the pubic symphysis, which is going to help with delivery, and it's also going to help make your ligaments very flexible in the sacroiliac, which is again in the hip regions, and the sacrococcygeal joints, again in the lower hip regions, and also helps dilate the cervix during labor. So relaxin is going to be involved in the late stages and in delivery. HCS is also known as placental lactogen, and lactogen, you know, with the word lactate, which means to make milk, and it's going to be helpful in preparing the mammary glands for lactation and also regulating metabolism in both the mother and in the fetus. We also secrete corticotropin releasing hormone, or CRH. It's normally secreted by non-pregnant people, so it is a hormone that's normally in circulation, but it's in much lower levels, and it's going to be secreted by the hypothalamus, and that's going to help with the timing of birth. So it's going to help with induction of birth itself, and it's also going to increase the secretion of cortisol. In fact, that's the primary role of CRH, is to increase cortisol secretion. And as I mentioned previously, that's going to help with stress levels, but in the fetal lungs, it's going to help produce surfactant, which is going to allow them to become mature. So it's going to help with those last stages of maturation of the fetal lungs prior to being able to take its first breath on the outside. The uterus is going to expand the entire time throughout the pregnancy. In fact, a typical way in which you can do a quick and dirty at-home test of how far along a woman is to feel for the fundus of the uterus, and it increases by about 1 centimeter a week. It's going to continue to expand upward through the pregnancy until it hits the belly button and then move itself up into the abdominal cavity until it fills it almost entirely, which is going to be quite uncomfortable for the pregnant woman because all of the organs are going to be smushed all over. They're going to be pushed out of the way and distended, and that's going to cause pressure on the stomach. It's going to cause pressure on the bladder, and the stomach, what can happen is you can have the food being displaced out of the way, and that can cause heartburn. You can also have stomach acids kind of coming up, so you can have issues there with heartburn at late stages of pregnancy. As you can see, this is the uterus. Behind that we have the fetus, which is about to become an infant because we are very close to delivery. And these are all of the rest of the maternal organs that are supposed to be occupying this space. So these here small intestines, descending colon, so we have the ascending, the transverse, and the descending colon, all of those would normally be in this region. Another thing of note here, this here is the urinary bladder. You're actually sitting directly on top of it, infant. So what happens in late stage of pregnancy is that a woman is constantly relieving her bladder because she constantly has serious pressure on it from this developing fetus. Additionally, pregnancy can make you uncomfortable in a lot of ways, including getting morning sickness, and fatigue, excessive fatigue. You also are going to obviously have an increase in your weight and severe posture changes because you're not just having an addition of the weight, but you're having the addition of the weight in awkward ways that can distend your back and your joints and your shoulders. And additionally, you've added to the mix increased relaxin, which is going to help allow all of your joints to become flexible. So now you have added weight in awkward directions, which can cause a change in the gait in the way that pregnant women walk. It's kind of a pregnancy waddle, they call it. But don't worry, eventually it ends. You end up with labor and delivery. And labor is the process of expelling the fetus. Generally, that happens through the uterus and through the vagina. If we have a C-section, we generally try to avoid the process of labor itself because there's no need to use muscles that aren't going to be cut through in a little bit. Labor is actually going to be initiated by multiple different hormones interacting and basically playing off of each other. And it's one of the very few times in biology that we talk about a positive feedback cycle. So many of the times that we've talked about the feedback cycles, we've talked about negative feedback loops, where we want to restore things back to homeostasis. But in this case, we have a positive feedback cycle where we want to continue perpetuating the condition until it resolves. So we have two different types of labor. Anyone who's ever been pregnant knows that false labor occurs quite a while actually before true labor, and false labor is associated with irregular contractions that are not they're not at timed intervals, and there is no blood or mucus or anything that's coming out of the vagina. True labor, on the other hand, is when uterine contractions occur at regular intervals and is going to actually hopefully result in the expulsion of the fetus, which then becomes an infant. And the first is the stage of dilation, whereby you're going to end up with larger and larger, so you'll actually measure the cervical dilation. And then the next stage is going to be the expulsion, when they say push, and you're actually pushing the baby out. And then the third is the placental stage, or when the afterbirth is going to be coming out as well. So the first stage is when we're dilating here. So this here is the vagina, this here is the cervix. So you can see the cervix is going to have to open up first, and then eventually the baby is going to come through the cervix and then out the vagina. Here we can see this little, I'm not sure how to describe it. It's actually the amniotic sac. It's going to be what's left of the amniotic sac after the baby has burst through this. This is when the baby bursts that amniotic sac, that's known as your water breaking. And typically this is a stage that a woman is going to be entering into labor and delivery very shortly. In fact, it's almost vital that she does so, because without that amniotic fluid, this fetus is at high risk until it actually has come out of the mother. It's at high risk for a lot of different things, including infection, if it's going to be in there without any of the amniotic fluid. But I digress. So here comes the fetus out of the cervix, and it's going to end up being expelled out of the vagina. Usually head first is ideal. If it comes out butt first, it's called breech, and it causes a couple of complications. Some other complications that people have to look for, or the physicians will have to look for, is the location of this umbilical cord, making sure that it's not wrapped around the joints or around the neck. Once the fetus, which again now has become an infant, once the infant is expelled from the vagina, we now have that last stage, which is the placental stage, whereby the placenta and the other fetal associated tissues are going to have to be expelled out. This is considered afterbirth. All right. After delivery, it takes a while for the woman's reproductive organs to return to pre-pregnancy state and pre-pregnancy physiology. This is called the periperium. And this is generally when women will succumb to things like postpartum depression, etc. So there's a lot of hormonal changes that are associated with it, as well as physical changes, including the reduction in size of the uterus. The uterus is going to take several weeks to actually involute, which means pull back down into its normal location inside the lower abdominal cavity. And boy, is it a new world for that infant. So the baby is completely dependent on the mother for survival. It's also Quite a transition for the mother, because the mother is going to have to cater to the baby's every single whim. So the newborn body is going to function independently, although all of its needs are going to have to be met by the mother. At birth, the lungs are going to be able to exchange oxygen and carbon dioxide. Again, this is something that started to happen around the 6th month, but then towards the later stages, we ended up with more and more surfactants secreted. This is one of the reasons why a premature delivery can result in a long hospital stay, because they have to make sure that the fetal lungs are actually able to have oxygen exchange. In fact, they have a novel treatment methods now for very early premature births where they actually are going to put them into a liquid. So instead of breathing from the air, they're going to be breathing from an amniotic fluid replicate that will allow their lungs to further develop for a little bit longer. The respiratory rate of an infant is very, very high. It's approximately 4-fold higher than a normal, than about a 2-week-old. Approximately 45 breaths per minute, and it drops to about 12 breaths per minute. And they also have a very irregular breathing pattern and breathing rate, where they may go for many seconds without breathing and then breathe very quickly to kind of catch up, because their respiratory reflex hasn't entirely kicked into full gear. Some of the other adjustments that an infant has to make post-birth include changes to the cardiovascular system. So that foramen ovale is going to close. Remember, that's the region between the two atria that's going to allow liquid to flow back and forth. It's going to close and become the fossa ovalis in the adult heart or in the infant heart. The ductus arterius is going to close and become the ligamentum arteriosum. And we'll look at that in a second. That's basically going to be the ligament that's going to connect the two major regions of the vena cava and the regions of the vena cava and the aorta. And what was previously the umbilical arteries are going to fill up with connective tissue, and the umbilical vein is going to become a ligament that holds the liver. So what do all those things that I just said look like? All right, so this is before delivery and this is after delivery, and we're looking at circulation here. So here we have the foramen ovale, and that's basically going to be a region between the two atria that allows for the blood to exchange back and forth, so that during development, high pressure changes of a contracting and beating heart didn't do too much damage to the regions that were being created during development, because the fetal heart was actually not strong enough to be able to handle intense changes in pressure. But now that it's a developed heart, it is able to do so, and so that has closed off, and now circulation is not going to be. This has a little bit of slosh back and forth. There's no slosh back and forth there. I also want to draw your attention here to the ductus arteriosus. This here is going to be a region whereby blood could previously circulate and now does not in the post-birth, because this now becomes the ligamentum arteriosum. I also want to take a second and step back. What we're looking at here is all purple. And the reason that we're looking at purple here in the fetal circulation is because the maternal circulation is what's going to supply all of the oxygen, and it's going to go in through these umbilical arteries and then back out through the umbilical veins, right? We're not breathing anything in through the lungs, so no oxygen is coming in through here. So that's why this is all purple. But after birth, obviously, we're bringing in oxygen from the lungs itself, and the umbilical vein is going to become this ligament here, this ligament teres for the liver, and the umbilical arteries are just going to become connective tissue. So those are some of the major changes that are going to have to happen as the infant adjusts to circulation that's coming from the lungs instead of circulating oxygen that's coming from the maternal blood supply through the placenta. When we're creating milk, that's called lactation. It's created from the mammary glands. It's produced and ejected out through the nipples and then picked up by the infant. It's going to have all sorts of goodies in there, including sugars and different hormones, and also antibodies that the mother may have created during different things that she's been exposed to throughout her lifetime. Prolactin is going to be a hormone that's secreted by the anterior pituitary gland and is going to be responsible for stimulating milk production. We also have another hormone called oxytocin. You may have heard as the love hormone, and this causes milk to be released from the mammary ducts via something called the ejection reflex, the milk ejection reflex. And this here is the physiology of lactation. So it's actually going to be a positive feedback loop. Basically, the mother is going to the baby is going to suckle on the nipple of the mother. Apologize. The suckling baby is going to increase the touch sensations. The touch sensations indicate, okay, I now have a suckling fetus. That means I'm going to have what's called let down. So they're going to have these nerve impulses that come from those receptors and go to the control center. in this case, and often, the hypothalamus and posterior pituitary, which then increases the oxytocin in the blood. The oxytocin in the blood is going to have that letdown or that milk ejection. And the effector cells of oxytocin are going to be these myoepithelial cells in the mammary glands of this lactating female, which are then going to contract. When they contract, they're basically going to squeeze that milk out. It's actually quite a forceful milk ejection. And if we continue with this cycle, we're going to continue until we end up interrupting that cycle, such as the baby ceasing to suckle, falling asleep, or pulling off, and that's going to break the positive feedback cycle of lactation. So there's a lot of benefits associated with breastfeeding your infant. First and foremost, you're sharing your immunity. So the white blood cells that you have as a female picked up throughout your lifetime of exposures to different things are going to get passed on, giving your infant an immune boost over an infant that doesn't have it. So that's those antibodies we talked about here. It also is going to be ideal for baby's brain development, growth, and digestion. And additionally, breastfeeding as a odd interaction where what the baby needs, the mother makes more of. So the mother's milk is not always the same. It depends on several factors, including what the mother is eating, etc., but also including what the baby needs. So a baby that is sick is going to have milk created by the mother that is a different chemical composition than when a baby is not sick. So the chemical composition of the mother's milk is actually going to change based on the baby's dietary needs. Additionally, breastfeeding is going to help support that optimal infant growth, because during times when the baby needs more nutrients because it's going through growth phases, the mother is going to detect that and her body is going to create more of whatever it needs. And it's going to lead to a reduction of several different types of diseases. And a lot of these studies are kind of biased because a lot of them were done quite a while ago, because we're looking at long-term effects of breastfeeding versus formula feeding. And if we're looking at formula from the 1970s and 1980s, it is drastically different, much more similar to actual breast milk here in the modern era. So a lot of those studies are a little bit biased because the formula that they were using wasn't exactly up to modern day standards. So, well, we've come to the end of our lecture. Well, thank you guys so very much for sticking with me. I appreciate it. Have a great day.